Liger is a hybrid cross between a male lion and a female tiger. Today ligers exist only in captivity because the habitats of the parental species do not overlap in the wild. Males reach a total length of 3 to 3.6 meters, meaning they are larger than large Siberian tiger males, who are themselves usually larger than the larger lion species. Israeli scientists have created a mostly featherless chicken by crossbreeding a broiler with a species that has a featherless neck. The idea behind the development of this ugly animal is that it will create a more convenient and energy-efficient chicken which can live in warm countries where feathered chickens don't do well and cooling systems are too expensive to be commonly affordable. Changes do not benefit the animals, and are in fact likely to make their lives worse. Males have been unable to mate because they cannot flap their wings for balance, and this also affects the females. Because they are featherless, chickens of both sexes are more susceptible to parasites, skin diseases, mosquito attacks, temperature variations and sunburn. Wholphin or wolphin is an extremely rare hybrid born from a mating of a female common bottlenose dolphin with a male false killer whales. They are extremely intermediate between both parents. Since a bottlenose may have about 88 teeth and a false killer whale has about 44, a wholphin will have 66. They are smaller than a false killer whale but are larger than a normal bottlenose. It is also assumed that a healthy wholphin may have a lifespan of around 40 years and grow to an average size of 12 to 20 feet, which are also a median between its parents' age and size. Though reports have been made about wholphins possibly living in the wild, there is not enough quality information or DNA tests that exist to give a conclusive answer to whether or not wholphins live outside of captivity, and if so, what type of habits and behaviors they possess. Bricanti mouse was a laboratory mouse that had what looked like a human ear grown on its back. The ear was actually an ear-shaped cartilage structure grown by seeding cow cartilage cells into a biodegradable ear-shaped mold and then implanted under the skin of the mouse. Then the cartilage naturally grew by itself. It was created to show that the transplantation of fabricated cartilage structures into human body is possible. GloFish is a patented and trademarked brand of genetically modified GM fluorescent fish. A variety of different GloFish are currently on the market. Zebrafish were the first GloFish available in pet stores, and are now sold in bright red, green, orange, yellow, blue, and purple fluorescent colors. In 1999, Dr. Haiyuan Gong and his colleagues at the National University of Singapore were working with a gene that encodes the green fluorescent protein GFP, originally extracted from a jellyfish, that naturally produced bright green fluorescence. They inserted the gene into a zebrafish embryo, allowing it to integrate into the zebrafish's genome, which caused the fish to be brightly fluorescent under both natural white light and ultraviolet light. Their goal was to develop a fish that could detect pollution by selectively fluorescing in the presence of environmental toxins. The modern version of the breed was developed in the 1950s by Professor Hansett, who worked at an artificial insemination center in Liege province. The gene mutation responsible for the breed's heavily muscled stature, more on this later, was maintained through line breeding, to the point where the condition became a fixed property of the Belgian Blue breed. Belgian Blue cows are more prone to dystocia, difficult birth, even when bred to normal beef bulls or dairy bulls, as they have an inherently narrower birth canal. In addition to the dam's reduced pelvic dimensions, the calf's birth weight and width are increased, making labor even harder. For this reason, cesarean sections are routinely practiced. He is the world's first transgenic dog. A clone beagle, Ruppy, and 4 other beagles produce a fluorescent protein that glows red upon excitation with ultraviolet light. Ruppy was created in 2009 by a group of scientists in South Korea, led by Byung-Chun Lee. The dog was cloned using viral transfection of fibroblast cells with and expresses the red fluorescent gene. Puppy was then shipped to the north of Croatia where it was thoroughly examined by Dr. Lisa Daichi, who clarified that the puppy could in fact be classed in the dog species. Even though it had not been born 100% naturally, it did still have all the characteristics of the species. This study is the first example where a four-legged animal has been bred to be see-through, but the reason for the transparency is relatively simple. The explanation for this phenomenon is that the new frog lacks pigment in its skin. Pigments are the material in animal skin, including human skin, which gives color. The amphibian cells that contain pigment, called chromatophores, are found in much lower numbers than the see-through brown frog. These cells are particularly sparse on the underside of the frog, making its internal structure very clear. Chromatophores are found in a wide range of animal groups, including amphibians, fish and crustaceans, but are absent in fish and birds. The reduction of these pigment cells in the frog's skin results in us being able to see through and into the animal's body. Whilst this study is fascinating in itself, it also has a range of applications in the fields of biological and medical research. The opportunity for a researcher to observe what is happening inside the frog's body eliminates the need for dissection. So, a researcher would now be able to study how the organs develop and change within the frog, without having to harm, or kill, the subject. Zebroid is the offspring of any cross between a zebra and any other equine, essentially, a zebra hybrid. In most cases, the sire is a zebra stallion. Zebroids physically resemble their non-zebra parent, but are striped like a zebra. The stripes generally do not cover the whole body, and might be confined to the legs or spread onto parts of the body or neck. If the non-zebra parent was patterned, this pattern might be passed down to the zebroid, in which case the stripes are usually confined to non-white areas. Mosquitoes are created for sudden and quick death. These mosquitoes are released to mate with the females, which in turn release the lethal genes from the male's mosquitoes and are transferred to the females, which kills them or kills the young ones in her, so that they can't reproduce. Scientists at British biotech company claims that it can control the spread of the dengue fever. It involves irradiating male insects, causing mutations that make them sterile, claims the company.